Our next speaker you heard from yesterday, but he's different today because yesterday he was Professor Purita and today he's coming to you as Dr. Purita. And we're going to talk a little photobiomodulation.
Thank you, Sean. All right, are we ready to rock and roll? A lot of slides again. And again, you can get a copy of these slides. So what we're going to talk about is basically photobiomodulation. This talk basically touches almost every talk that's been given in this meeting because it's pertaining to everything we see. So what is it? Basically, it's involving low-intensity light to affect various cellular functions, pathways, and basically promote therapeutic effects. So we can see here how it works. Now, there's some very important things here. If we look at the primary effects of photobiomodulation, we see it's calcium. I said, wow, calcium. Then we have reactive oxygen species, ATP reduction, nitric oxide production, pathway influence. So I said to myself, what about the calcium signaling pathways? Well, they're of paramount importance because basically they really will regulate all sorts of pathways in the body. So it's so important, and photobiomodulation really affects these well. So we can see here the various aspects of photobiomodulation: angiogenesis, collagen synthesis, ATP production, mitochondrial health, pathways, and cytokines, exosomes, etc. So you can see. Now, here's some of the basic science. We've got to do this. You've got to know it if you want to know this field well. So you have ionizing and non-ionizing radiation. We're basically interested in the non-ionizing radiation. That ionizing one is what they use for cancer therapy, things like that. So we can see here the various types of electromagnetic radiation, which this all is. But we're basically going with the visible light. Now, you might remember from your science days in high school, Roy G. Biv, red, orange, yellow, green, blue, indigo, violet. That's the colors that we're really working with in this case, okay? And we can see here on this particular slide how the various colors penetrate into the cells. You know, you're only going to get a certain depth of penetration here, but you can also stimulate things by, getting that depth of penetration. So I don't want to, get into too much between lasers and LEDs and everything. I'm just giving you this for some general concepts to kind of understand what's going on. We could give a lecture just on that alone, certainly, okay? So basically, what are the physics of light therapy? You have wavelengths. Okay, a wavelength basically determines the light, color, and energy. You have the frequency and you have the amplitude. Let's look at the next slide. That kind of explains it a little better. So we have our wavelength, we have our amplitude, and we have basically our frequency. So let's get into that a little more, okay? So when we talk about frequencies, and this is where science is really going to start going to. It's frequencies of compounds, frequencies of cells. So we have a frequency, how many pulses of light there is per second. And then we have the duty cycle, is how many pulses are on and off in that second. Now, I don't know much about this field, but I can tell you in another year I will, because I'm really going to start learning about the frequencies of pulsing, because I think that's so important in how things really work. So you can see some hertz here. This is how much that light pulses on and off, And here's some other ones, some other frequencies of various aspects. And again, these slides are available to you, so don't go crazy trying to write down these names. But we know for sure pulsed light is much more efficient than continuous light. It has a much greater biologic efficiency, and you can see here what it does. So now we get into some of the other aspects of photobiomodulation. Optogenetics. Basically, light-sensitive proteins in the body are called opsins, okay? And we can see by the bottom there, you can see how when the light is on, it stimulates that nerve cell to start producing all sorts of things. Okay, so basically when we start looking at the wavelengths, we have the photoreceptor activation and the transduction pathways. Again, this is kind of boring, but I'm just going to, zip through this part. But we can see here you have cryptoprotochromes and things like that and phototropins. But let's get into a little more. So the chromophobes. Okay, so basically, and you're going to start saying, oh, wait, this is starting to make a little sense now. A chromophobe gets excited. It starts to absorb that energy, and then it transfers that energy to another molecule. Now we can start seeing some things. We start looking at, various aspects of various color lights, blue light, red light, green light, and things like that. And we can see how it starts affecting certain pathways. Okay, so now we start seeing things. So the chromophobes, I should say, convert the light energy into chemical energy. And when it absorbs a photon, it's called an exciton. This picture shows you that there. So we have them in the bottom. We can see the valence band. and then all of a sudden that, photon gets in there and it gets that electron to go up, and next thing you know, we have some transfer of energy. Now we start talking about cytochrome C oxidase. It's a chromophobe. You've heard about that. Electron transport chain. We said, well, hell, we don't need to know about that. Well, let me tell you something. That's so important in regenerative medicine, stem cell therapies, et cetera, because so many diseases are dependent on that electron transport chain. And you can see here what it does is it helps to regulate the reactive oxygen species and things like that. So cytochrome C oxidase is very important in the electron transport chain. And basically red light stimulates it, okay? So that's why we like to use red light in a lot of our stem cell therapies, because you're stimulating that cytochrome C oxidase, you're going to get more energy produced, okay? Now, most of the lectures also that we're dealing with in this meeting are eventually going down to the health of the mitochondria. And we know that harnessing light energy is going to help mitochondria. Dysfunctional mitochondria can't effectively convert light energy into ATP. Remember, mitochondria is sort of like a chlorophyll, or chloroplast, I should say. So many disorders are basically energetic problems. Now, you can see here, it's cytochrome C oxidase is a critical regulator of oxidative phosphorylation. It produces the energy. Without it, we're going to have trouble. So it's also very important in cell signaling pathways. So it's so important. And a lot of diseases I think we're going to find are going to be related to cytochrome C oxidase. Now you can see here, here's how it works here. This is the electron transport chain. That beam of light goes there. The electron transport chain gets activated, and all of a sudden now we're making more ATP, and that, cytochrome C oxidase is basically in the mitochondria, in the inner membrane, and it's basically helping to get those transport of the electrons. Now you can see here where it is, between 3 and 4. Now why is the electron transport chain so important? This is a very profound slide. This explains most of your neurodegenerative diseases. You can see here now where these diseases lie. Some of them in phase 1, some phase 2, some phase 3, some of them in all of it. Most degenerative diseases come down to basically electron transport chain and problems inherent in the chain itself. That's why basically we may have some hope with photobiomodulation. Okay, so basically photobiomodulation increases nitric oxide. Nathan was talking about that, how important that is. How do we increase it? Well, photobiomodulation can increase it. It photodisassociates nitric oxide from cytochrome C oxidase, increases our nitric oxide levels, which is so important. And I have a good slide here. It shows it there. So basically, it produces 4 protons, which go on to help form ATP, the cell energy. That's the energy currency of the body. Okay, we can see the same thing again. What happens, that nitric oxide gets basically torn away from the cytochrome C oxidase. It does a lot of good things here. It's a game of, of numbers, and this is what helps. All right, so basically same thing. I'm showing you the nitric oxide production. Okay, so basically another thing that was mentioned, I think, earlier, they're sirtuin enzymes. So important. How your sirtuin enzymes work is how you essentially are healthy or not. And photobiomodulation has a very important aspect in controlling them. And here's what the sirtuins look like. Look at all the things that they control, 1 to 7. Some are found in the mitochondria, hence photobiomodulation. Some are found outside the mitochondria in the cytoplasm of the cell. And basically, another interesting aspect, I was talking to Bill Landers about this yesterday. They help increase and stabilize telomerase, okay? And this is the aspect of how they do that. Basically, they're going in there and they're stabilizing something called the chaterin proteins. So we're now knowing that photobiomodulation can also help telomerase. So we start seeing all the things, okay? We were talking about telomeres. We were talking about nitric oxide. We're talking about stem cells. All of it is coming into play here, okay? Now, another very important aspect of this is it helps decrease oxidative stress. Another big thing that, how many lectures have we talked about? ROS. So basically, how does it do this? Well, it starts inducing transcription factors. NRF2, it hangs out in the cytoplasm. Photobiomodulation releases it from the cytoplasm. It goes into the nucleus, gives those genes instructions to start producing antioxidant enzymes. Okay, nitric oxide, we talked about it, but you realize now one thing. Nitric oxide can be your buddy, but it can also be your enemy. A lot of people that had cytokine storms during COVID, they had too much nitric oxide. So as I put in red there, it can act as an antioxidant in certain levels, or it can cause significant oxidative stress. Goes back to that big principle we talked about, hormesis, hormesis, hormesis. So it also basically has importance, and it can promote cell survival pathways also, which I mentioned here, mTOR, things like that, okay? Now, hypoxia-inducible factor, that was mentioned a little earlier when they were talking about hyperbaric oxygen. This is one of my pet interests, you know, intermittent hypoxia, things like that. So let's look at that. You know, so what happens is the photobiomodulation gets the hypoxia-inducible factor, which again is a transcription agent, hangs out in the cytoplasm, goes into that nucleus, and look what it does. Angiogenesis, erythropoiesis, apoptosis, cell proliferation, so many good things. Again, photobiomodulation. It's kind of like I'm just following up all these other lectures for that. Okay. Now, how about stem cells? This is a stem cell meeting when all is said and done. Okay, so it can help stimulate stem cell proliferation and migration, upregulation of growth factors, moderation of signaling pathways, and again, ROS, regulation of the reactive oxygen species. So basically, again, it has the activation of transcription factors, modulation of epigenetic mechanisms. You know, epigenetics is the number one thing that we're going to be dealing with, I think, as time comes on. How we turn genes on and off is going to make difference between success and failure, life and death, okay? So that's the way it goes. And again, regulation of the signaling pathways. Now, we can see here, this is some things where we have osteoblast, and you can see here how it goes to osteoblast and basically mature osteoblast. So it can help differentiate cells also, not just make them healthier, but it can kind of guide them towards going to differentiation. That'd be usually a blue light, things like that. Another slide that again shows the fact of photomodulation in stem cells. Okay, so how does it affect the wound healing process? It helps produce collagen, which is the basis of everything. Remember now, collagen is found in your extracellular matrix. That's really where a lot of problems lie also, in that extracellular matrix between the cells. You know, if that matrix is not healthy, you're not healthy yourself. Okay, so it increases collagen type 1 and type 2. The wavelengths between 630 and 830 seem to help collagen production. So that's kind of in the red light, infrared light range. Okay, so we can see here what photomodulation does. You can see the various aspects, wound contraction, this and that. So yes, you can put exosomes on. Yes, you can put stem cells on. You have a nitric oxide cream. But you still have the photomodulation to really put everything together. Here's another good slide that really kind of tells you wound healing in the shell. You have primary intervention, cellular changes, cellular response, and eventually tissue changes. So this can make a tremendous difference, and it's really not that expensive, and certainly not invasive when all is said and done. So it's really a good thing to utilize. Okay, now, how about the mechanisms of photomodulation in the brain? Well, you know, I have a slide here that you could probably spend 15, 20 minutes discussing this slide. But basically what it does is increase antioxidants, does increase the anti-inflammatory compounds. Basically, it also increases neurogenesis. So all the things we want to see when we want to try and get a brain injury better. And I know some of the people, when they do, for instance, hair treatments, Dr. Bauman puts a red light hat. You know, it looks like something from Star Wars, but it seems to work. So there you are. And then, again, some of the things of photomodulation, stroke, we know it works with that. Traumatic brain injury, spinal cord injury. I'm working down in Brazil on that. We're having some pretty interesting results in that. I have another slide that I'll show you shortly on that. Neurodegenerative conditions we talked about. Also anxiety and depression. Certain wavelengths of light can actually help depression. I think it comes a little bit later in the talk. And here we are here, effects of photomodulation in neurodegenerative conditions. Very multifaceted, anti-inflammatory, anti-apoptosis, basically decrease in plaque formation, things that we've been talking about in other lectures, but it kind of helps come together here because this kind of solidifies things. It's the glue, so to speak. Okay, what I was talking about here, this is an interesting article. This is an article that just came out about 5 weeks ago, how basically they implanted a little stimulator with light for spinal cord recovery. There's a lot of literature out there how this is so good for spinal cord injuries. We're basically using a combination down there of bone marrow aspirate, and we're getting pretty good results. We're putting it intrathecally. All right, so photodynamic therapy, another kind of therapy that we're talking about. So we have a photosensitizer. We can go after bacteria. We can go after viruses. we can go after cancers. So very interesting therapy. So let's kind of get into a little more of the nitty-gritty of it. So basically, it's beneficial also for not only just destroying those organisms, we get the 1-2 punch, because not only is it getting after viruses or cancer cells, it's also increasing mitogenesis and mitophagy, okay? Because it's causing a slight reoxidative stress. And that oxidative stress is what kind of helps those mitochondria recover better. So you can kind of see it here, okay? So we have little Dorothy from The Wizard of Oz. And this is kind of the principles here. You can see the photodynamic therapy, and basically we have various light sources. The sun is one of them, believe it or not. And then we have the photosensitizers. What does it do? It creates reactive oxygen species. A normal cell can usually kind of withstand that little bit of stress, but a cancer cell or a cell that's kind of invaded with a virus can't withstand it, and voila, the cell dies. So that's one of the things we like. And at the same time, we're hopefully getting mitogenesis and mitophagy. Good things. Now, this is a new technique. This is literally the first place that this technique has ever been looked at. And you know, one night I was kind of laying in bed. I said, what am I going to call this technique where we're starting to activate compounds with our pure light? I said, well, okay, I'm going to call it EPAT, extracorporeal photoactivation therapy. You can see on the right there are some of the compounds we've been photoactivating now. We're kind of fooling around. I'm not going to tell you we've got it down pat. We're learning. Okay, I just did an NAD on myself, you know, about a week or so ago, but it worked pretty darn good. So these are some of the compounds we think are going to work. So let's get into these a little more. Methylene blue, photosensitizer, and also a mitogenesis stimulator. Very important, okay? So it can react with a lot of components of the electron transport chain. And we can see it here in that cytochrome C oxidase again. Now you can see the importance of this electron transport chain. It's not some esoteric thing that just is in the lab with the PhD guys. It's practical. It's important. Okay, so there you are. So methylene blue. It affects cytochrome C oxidase, makes it more effective. More effective cytochrome C oxidase, more ATP. So that's why I like that. So it's also very good for viruses because it gets accumulated in the liver, places where the viruses kind of hang out. What we were doing until just a few weeks ago is giving patients methylene blue and putting them on a red light bed then. Now I said, well, forget that. Now we're just, you know, photoactivating it ahead of time, giving it in an IV bag, and boom, it's done. Be careful with methylene blue, though, if you have a patient on an antidepressant, because it increases serotonin levels, which is sometimes a good thing. Again, we talk about depression. Now, methylene blue and mitochondria. I was just talking to Nathan about this yesterday. You know, there's some people out there that say, well, methylene blue is going to really make the nitric oxide not work well, and it's going to make your mitochondria not work well. It's a controversial topic, but I did a deep dive in it, and I think it's really not the case. So you can see here, here's some of the important aspects of the methylene blue, some of the dosages that we use, and again, it's affecting the cytochrome C oxidase. All right, so basically, it's a potent inhibitor of nitric oxide signaling in some respects, but what we think it's probably really an inhibitor of inducible nitric oxide synthase, which is fine if you have a bacterial infection, but with regeneration, we don't care. We don't want to have that increased. So it doesn't really affect the endothelial nitric oxide synthase. But here's the bottom line. Cover your bases. Take a nitric oxide supplement. This way there's no question. Boom, done. All right, so again, it can really affect the mitochondria. I'm trying to really speed along, see if we can get to the things. Here's a cute little article about that. And curcumin, another good thing that we use with photobiomodulation. Pretty much what I said about methylene blue holds for curcumin too, except we use a violet light to activate it. Now, I'm still trying to figure out what frequencies I'm going to use to activate it as far as pulsing on and off. We're going to give it time and see, figure it out. But there's some of the things about curcumin. All right, so basically you can see a lot of antiviral effects. So probably I'm going to start using them both. I don't think at the same time. I'll use one, maybe another day I'll use another, okay? So there's some of these pathways. NRF2, important pathway there. Again, NAD. Now, I've talked to a couple of people. They were doing it with red light and having pretty darn good results. There's a clinic down in New Orleans, or not New Orleans, nearby there, the Spring Hill Clinic, and they apparently have been using some NAD-colored light, somebody that's working with them. Seems to work pretty good, so we'll see. The jury's still out, but that's what we go there. Now, green light basically can affect certain aspects of our circadian cycle and things like that. And it can also do some good things. And again, you can look at this. I just have something there for red light. And then photomodulation and arthritis. We know it makes joints feel better. We can tell you in our office, sometimes we'll see the patient the week after we do something. They're still sore. We give them some red light. They're much better. All right, blue light, basically similar things. Certain pathways that it'll stimulate. I'm going to see if I can go just a little bit more. Interesting thing, blue light stimulates the production of exosomes. Another topic that's obviously been very popular at this meeting, okay? And ultraviolet light, what does UV light do? Well, that may stimulate heat shock proteins. Again, it's a mild oxidant, a mild stimulator of causing some inflammation, and it stimulates heat shock proteins. And believe me, heat shock proteins are so important in our field. And then you can see here, P53 is one of the heat shock proteins things. Now, I'm going to end up with this, macrophage activating factor. There's something called negalase. It's a protein produced by Lyme disease and things like that. And basically what it does is it turns off our macrophages from working. We're looking into that. We think photomodulation is a way of taking care of this problem. And we have a couple of other things that we're working on. But this is what it looks like here. And it's basically if you get your immune system working better, and this basically is paralyzing your immune system, this is going to work better. And we think that this is how it works and how photobiomodulation will take care of it. So anyway, thanks so much, guys. It's been my pleasure. And Sean, it's an honor to work with you guys.